Wait, let me put it up on screen. So this is human brain organoid assemble functionally integrated bilateral optic vesicles. Um, and basically, so you guys you know what a brain organoid is, right?
Yes. Well, I know. I don't know who is watching, but the organoids is what scientists now can make from stem cells, right? And it's used as an alternative to studies in animals. Also, like it's you kind of grow an organ, but it's not an organ, so you call it an organoid, and then you can test this and look what it does.
Yeah, I think that's a perfect description. You grow an organ. In this case, they make a brain organ. Um, so you can grow like a bunch of brain cells that all integrate, and then I don't know if they have blood vessels in there. That's usually the limiting factor. Like there's no, it lacks structure. So most of these
Mhm.
organoids are very small because you cannot make the big ones because then you can't get nutrients and stuff in it because you don't have a heart. You just have a blob of tissue in a dish.
Yeah.
Anyway, do you want to see something mildly disturbing?
Oh yes, I would love to see something mildly disturbing. I don't think it will be very disturbing.
There you go. It's an organoid with eyes.
Oh no. Oh, okay, that is mildly disturbing.
Right?
Yeah, that is mildly disturbing.
um,
So this is what they meant with optical vesicles, or what did it
yep.
say? Is it something about optics and something about vesicles? So
Yep.
organoid with eyes. This is what it means.
Basically, yes.
Whoa.
So it's a human brain organoid with a pair of pigmented eyes. They use retinol to induce the formation of, on one pole of the organoid, two eye-like structures with a cornea, lens, and a light-sensitive retina.
Oh wow, it's actually an eye. Like it tries
Yep.
to be a real eye.
It responds to light and everything. Um,
Oh, so cool.
right?
Okay, but it's induced. They have to induce it somehow, right? Because you start out with kind of stem cells and you induce what kind of organoid you want. And here
Y-
they induce the organoid to grow eyes with some substance.
yeah. So this is kind of the whole developing field now in iPSCs. Um,
Mm.
so they are induced pluripotent stem cells. So these are not necessarily stem cells taken from embryos or whatever. These are literally just you can take skin cells and then you can revert them back.
No.
So every
And
cell starts off like we do with infinite possibility.
yeah.
And then as we mature, we narrow and narrow and narrow. Cells do the same thing. So our cells will start off being capable of turning into an eye or a brain or a blood vessel or whatever. And then eventually they turn into that. And we very long thought that you couldn't go back. And then a couple of decades ago, people realized no, you can go back to that stem cell state for each cell. So
You
that's
can
been
do whatever you want. Even when you're old, you can go back and go another way.
exactly. So we've gone back, and from that point onwards, it depends very strongly on what kind of environmental factors you provide these cells. And we've worked with some labs that were making muscle neurons, motor neurons, sorry, muscle cells and motor neurons. And then there's actually a very expensive mix of drugs you need to apply because these are like hormones and stuff that you need to apply at the exact right time in the right concentration. And then these cells are like, oh, I'm supposed to be a motor neuron. Very well then. So figuring out the environmental factors necessary to turn a stem cell into useful tissue has been an ongoing field, very active, very interesting, and um, I don't know if this has been done before, but the way it's been brought is, let's put it up again. So basically, they found that if you apply retinol, which is normally found in the retina, hence the name, then those stem cells will turn into eyes or eye-like structures.
I was
So there's a
thinking,
study here. Human brain organoids assemble functionally integrated bilateral optic vesicles. Um, I don't know exactly how they did it. It's very far outside of my field, but
yeah, but do you know if you need to apply this locally somehow, or do you apply this to this retinol to the whole organoid and then it grows exactly two eyes? Why doesn't it
I
grow 10 eyes?
assume this is applied to the whole organoid because these
Yeah.
organoids are very small and they're typically kept in a dish with nutrients and oxygen and
Yeah,
stuff
it's
to keep
just
them alive.
one dish, right? You don't
Yeah,
have a
it's a blob
separate
of
flow
flesh.
for, yeah. Yeah,
Let me see if it's anywhere in the summary.
that's pretty cool.
Using induced pluripotent stem cells, so these were normally normal cells, and then they were turned back into stem cells, and then forward into brain cells, and then including brain organoids. That using induced pluripotent stem cell derived human brain organoids, we attempted to simplify the complexities and demonstrate formation of 4 brain-associated bilateral optic vesicles, cellular diversity, and functionality. Around day 30, brain organoids attempt to assemble optic vesicles, which develop progressively as visible structures within 60 days. I don't. Interestingly, various light intensities could trigger photosensitive activity of OVB organoids, and light sensitivities could be reset after transient photobleaching. Wow. So part of this is also once you get a group of cells to start differentiating, they will start releasing the hormones needed to differentiate further and further. They will organize themselves. So I don't know if how they induced it. I didn't read the whole paper. This is just the summary. But essentially, once they start going, they'll just keep going and hey, I'm turning into an eye. Okay, then I need to release this hormone, and then the cells next to me will also turn into an eye. And biology is amazing, and like I said at the start of this, just very mildly disturbing.